When Lexi's seizures first began that were noticeable to us, I was very scared. I immediately knew that night that it occurred that something was wrong. I didn't know exactly what, but it was very clear that something was wrong.
KCNt1 is one of the most catastrophic pediatric diagnoses that a child can have. For many of our kids, it means having anywhere from 50 to 200 seizures a day. The seizures are not controlled in any way by the existing medications. And then on top of that, they have profound developmental delay. So for an example, my son, who's now 8 years old, functions like a 6-month-old. He can't walk, he can't talk, and he is 100% dependent on us for everything, every minute of the day.
The brain learns in two ways. It changes the strength of connections between one brain cell and the next, one neuron and another. And it also changes the way each brain cell, each neuron, responds to an incoming input. Ion channels are fundamental to how the brain works because they are the proteins that generate every electrical impulse in the brain. One of the most interesting channels that we have been studying is this channel that we call KCNt1. When it becomes active, it actually instructs the neuron to start making new proteins. And they generate also the impulses that then go out to your muscles that produce every type of behavior that you make.
The way mutations in KCN affects the genetics and the patient is that it changes the way the actual channel, this potassium channel, works. Potassium is an electrolyte that has a charge, and the change in the charge helps send impulses that allow the nervous system to work. When that channel isn't working properly, when the different charges aren't flowing as they should be, it causes the brain to have a malfunction that can lead to problems with both learning and development as well as seizures.
So whenever we engage in learning, there are going to be changes in the neurons of our brain, either strengthening or weakening of the connections between two neurons. And this is required for the formation of that memory. When we want to later recall that memory, a process called activity-dependent translation or the creation of new proteins is required through various mechanisms to help consolidate that memory and make it available for us to then recall it when we need it. So whenever you have dysregulation of the processes required, either the immediate processes when the strengthening or weakening of the synapses or this activity-dependent translation, you're going to have problems with development because you're not going to be having learning when the appropriate external cues are coming. And whenever you get dysregulation of this process, you're just going to be losing the ability to form and then meaningfully retrieve these memories later in life.
In most cases, these children will be identified because they're having seizures very early in life that the parents see or other family members see. But in some cases, the subclinical seizures are actually something that start to occur much before any clinical seizures do. Some children may mostly or exclusively have subclinical seizures, which we wouldn't know about without EEG monitoring. In those cases, the presentation would be that the child presents with developmental delays, late milestones, and then we would find out that they're having seizures afterwards.
So KCNt1 is a sodium-activated potassium channel that helps regulate neuronal excitability. We discovered that KCNt1 doesn't work alone. It physically interacts with proteins that regulate RNA translation, especially FMRP. So this suggests that KCNt1 can directly couple changes in channel activity with the RNA translation in neurons. To understand the underlying mechanisms and guide the treatment strategies, our lab generated a mouse model carrying R455H mutation, which corresponds to R474H in patients. So using this mouse model, we performed the patch clamp recordings from cortical neurons. And we find that this R455H mutation persistently activates KCNt1 channels and increases both potassium and sodium currents. These recordings are important for understanding learning and memory because they show in real time how the KCNt1 activation alters potassium currents and neuronal excitability. By linking electrophysiological properties with the translational regulation, we can see how changes in channel activity were converted to cap-dependent translation or protein synthesis that supports synaptic plasticity, learning, and memory.
The reason it is so important to continue to invest in early discovery science is it is the backbone to treating diseases better, to reducing people's suffering, to reducing healthcare costs, to improving long-term outcomes, making more people be able to be healthy and living their best lives.
One important thing about finding a genetic diagnosis is that it ends this very difficult and protracted diagnostic journey that the family has been down trying to understand what's happening to their child. Secondly, it helps us in some cases to identify precision treatments. In the case of KCNt1, this was initially led to efforts to try newer small molecule treatments and also antisense oligonucleotides. In many cases, it helps us to funnel these children into clinical trials that may be a benefit to them, but really just the knowledge for the patient's families is often the most important thing.
What gives me hope is watching the wins that we've seen in other rare communities. When Andrew was born, we were told that if he was for a therapy to be meaningful, that he would likely have to have that in the first year of life or maybe the second year of life, because his brain would be so tragically impacted by the hundreds and then thousands of seizures that he was enduring. But over time, we saw example after example of children at more advanced ages having profound improvement on drugs. So all it takes is a single example of those, and there's plenty of them out there. Let's say if that disease is amenable to a late intervention, and that a kid that is that profoundly sick and disabled can improve, then then ours can too.
My hopes for Lexi have changed throughout her life. Lexi was diagnosed before the KCNt1 Foundation even began. And so at this point in time, now that there is so much hope for research and treatments, my truest hope is that we can prolong Lexi's life in the setting of comfort and happiness. Anything that she could possibly accomplish throughout that would just be a bonus.
One thing that has always motivated me and our lab is just simply trying to understand how the brain works. It's only with a deeper understanding of how that happens that you can actually develop treatments for diseases. So everything we had already learnt about the channel, without actually having to think about the clinical implications, allowed us to very rapidly try and figure out what was happening with the mutations and how that was going to affect the normal function of the neuron. If we hadn't been studying it for so many years, we wouldn't have been ever in a position to discover what the likely cause of the intellectual disability actually is when you have these mutations. That has been an incredible motivation to try and fix the problem. We have interacted with patients and parents of patients, with foundations, and of course with biotech companies that are trying to find approaches to fix this. That has been extremely rewarding to see that we can actually do something in that direction.
Just look at that smile. Oh my gosh. He's the sweetest.